ultraviolet light. It's like a very intense sunbathing bed. I don't suggest you look at those lights. I've already done this. You put the UV on for 15 minutes to make sure that anything is killed in there, because sunlight will kill everything. It's why when you have, after you have like a flu or something, the idea is wash your bed sheets and then hang them out on the line if you can. And the idea is that sunlight does unravel DNA. And so, particularly in this hood, we're doing a lot of PCR, which is a way to replicate the DNA. So it's a molecular technique that we use, and we need to make sure that the only DNA we're actually working with is the DNA we want to be working with, and not stuff that fell off of somebody's hands or is a contaminant from another project. So that's what this is. I left my lighter in the other room. So again with the flame. Okay, so this has got air flowing this way and underneath. It comes in through a filter, and it's also pulling anything out. So you can see that the flame is actually bending in the direction of airflow, so that's just something you have to be careful with. I'm going to set this up to flame. This is what we call a loop. I've broken this one because I like to have a pointy end as opposed to a circular loop, but we do have those as well. And the idea is we want to get the end of this super, super, super hot. So I'm going to let that sit and flame for a minute. And let's have a look in the incubator. So this is just a chamber that keeps everything at 20 degrees Celsius. That's about the temperature your house is at. You know, it's warm. We have a lot of fun stuff growing in here. Stuff from other projects and stuff that we've been isolating out. At this point, most of the stuff in here is pure culture, and what we mean by pure culture is there is one organism and one organism only, one type of organism on each of these plates. I didn't mention before, but these are potato dextrose agar plates. Agar is a derivative from seaweed. In fact, you can eat it, and you do in a lot of Malaysian and Japanese cultures and things like that. All of the Asian cultures have this dessert that they make with like coconut milk, sugar, and basically agar. And it's like jelly. It's really cool. I've eaten it. It's okay. In fact, I've tasted PDA, and that's okay too. It's not great, but it's okay. You could probably live off of it, and fungi do. So it's called potato dextrose agar because it's made with agar, dextrose, which is a sugar, and potatoes. So you can make different types of agar. I've made in the past V8 agar, which is literally like V8 tomato juice and agar. And so we mix it up literally from a solution that we create using this stuff. You can do this at home with like literally just boiling up potatoes and agar. But this is basically just a powder. Is that open? Doesn't smell so great, but it's literally just potatoes and agar, and we autoclave it, boil it up, and then pour it into plates. That's really all there is to it. What we're going to show you guys is how to isolate things. I'm trying to find a culture that's actually kind of nasty and has more than one thing in it. Okay, so this one's obviously got two things in it. Your roots that we just did will have like 5 or 10 things coming off of each root. What we're going to do is we're going to take a sample from this pink one and a sample from the brown one and plate them out onto separate plates. So we go back over to our hood. At this point, I've just been messing around in the incubator, so my hands are filthy dirty again. I can change gloves, which I want you to assume that I just just have, and then I sterilize them again. I actually don't need to change gloves as often as I would if I were doing research, because this is going to get them clean again. And I haven't been in any dirt, I won't have anything really stuck to them. All I've been doing is touching surfaces. And theoretically the surfaces in here are pretty clean. We have HEPA filters in all of our air that's coming in here, again because we're doing molecular work and not just culture work. So again, bleach and ethanol. Often one of the biggest sources of contamination in a lab will actually be people's lab coats. I wash these in bleach every couple of weeks, but some of those lab coats, I know that doesn't happen. So, we're in the hood, it's kind of noisy here. What we want to do is get this probe white hot, or at least red hot, which we can accomplish in fairly short order. It's a very thin filament, it's going to get very, very hot. They say red is dead, and that's what is happening to these organisms that might potentially be living on my probe. When I'm just starting to get into the sterilization, if I haven't been doing it and I don't know what might be on this probe, I also want to heat up the ends of it. After I'm done, I'll set this in an ethanol bath as well, because nothing should be alive in ethanol. So that's clean. Parafilm off of this. Again, I don't have fingernails, so this isn't working very well. Fortunately, fingernails grow, so we'll be efficient again in a couple weeks. Okay, so these were from a project that this researcher worked on. She originally cultured these on February 22nd, 2015, so they're a little old, they need to be redone. And for some reason she called it Smelly Cat, which might mean that she cultured it off of a cat. Don't know. We'll just replicate that. And I'm going to call this isolate 1 and this one isolate 2. And I'll put the date that I'm doing this as well. Today is the 24th of August of 2015. So you see how we're tracking the data? We know who originally started the project, who originally cultured it, what it was from, theoretically. I could go and ask Abby, whose name that is. I could ask her what this is, and now we know who has recultured it. And this is all just part of record keeping, and that's important in science. If you don't know when something was done, who it was done by, or what project it was done for, you don't know what the science was. So, after we've sterilized our probe, this is really hot. If we touch it to the organism, we're going to kill it. So what we do is we actually dip it into the agar just to cool it off. And I like to wait about 10 seconds after it's done sizzling. That's just my rule. Then we're going to go to an area that is not contaminated. You see there's this interaction zone between the two. I don't want to pull any of the tissue from there because it could be this guy or it could be this guy. So what I'm going to do is I'm going to go way off here on the edge and snitch a little bit of the spores. You really don't need very much tissue. You probably can't even see what I've got on there. I'm going to poke that right down in the center. That's going to grow out and fill the entire plate. Again, make sure our loop is really clean. Cool it off. And we'll try to get this other one. I don't know if it's going to come up because this culture is a little dry. I might not be able to get that one. We'll try a bit of the fluffy stuff on the edge. So fungi are incredibly tiny, and you really don't need much material in order to get growth. And we're just going to toss this away in an autoclave bag. I will heat those guys up and kill them quite thoroughly later. And at this point we can turn the flame off and we'll grab some charcoal strips, just like we did before, and seal them up. Again, I want to stress like the minute amount of material that you actually need. There's a saying that one bad strawberry ruins the bunch, and that is absolutely true because the little bit of mold that might be on one strawberry could contain millions or even billions of spores. Fungi reproduce mostly asexually through spores. Their sexual structures are the fruiting bodies you know as mushrooms or morels. Most of the time though, they're just reproducing through spores or molds. So the same type of fungus can reproduce both through a mushroom fruiting structure and then through spores. Most of the time they're doing it through spores or just by breaking off bits of their hyphae, which is their vegetative body. The reason for that is it's really efficient to not have to go and find another fungus to have fun with. It's way more efficient to just be able to break off part of you and have that grow off and be a new organism. So, that is that. We'll close that up, turn that off, and I'm just going to set the UV to go again because I've had open spores in here, and just to make sure it's all nice and clean for the next person who comes along.